In this tutorial we will discuss the HTO demultiplexing module. Of course, before we can proceed with the demultiplexing, the project must have been loaded. As you can see here, the project is called FL Tutorial. It contains some 10,000 cells and more than 18,000 genes and 4 HTOs. The first step to demultiplexing is to normalize the HTO counts. Several methods are available. You have a helper to help you with the information. By default, we propose to use the center log ratio method, but other methods are available if needed. By clicking on the Normalize button, the normalization is started. When the normalization is completed, it is necessary to proceed with the demultiplexing itself. For this, two solutions are proposed: either use the Multisec Demux tool which will perform the demultiplexing automatically, or proceed manually with a quantile system. This is an expert mode. We recommend using the automatic method. Just click to start the process. Once the multiplex is done, several information are available to you to evaluate the quality of the result. First of all, the counting table. Here we have 4 HTOs whose names are 1 HUFLCE, 2 HUFLCE and so on. We have opposite the number of cells that have been associated with each HTO. Here we also have the number of duplicates, i.e. the number of cell code bars associated with several HTOs. Finally, we have the number of negatives, i.e. the number of cell barcodes that have not been associated with one of the HTOs. The first figure we see is a UMAP figure that has been calculated in the HTO space. Each point represents a cell and the color represents the association that was found by the tool. We see that the 4 HTOs form separate clusters. The blue duplicates form areas between two clusters of HTOs and the purple negatives are in a central area where it appears that there are not enough HTOs to analyze the original sample. The second tab allows you to access the ridge plot. The ridge plot is a representation of the signal captured by the demultiplexer. On the line you have the identity information provided by the tool, either one of the sample, doublet or negative. The title of the figure is the indication of the HTO whose signal has been measured. We observe, for example, that for the HTO, 1 HUFLCE, the cells that have been associated with this HTO, i.e. with the same sample, have a good quality signal, while the cells that have been associated with other samples have a low value in this HTO. The duplicates, on the other hand, have two signals, a low value but also a positive value since they can be associated with this HTO and another. This allows you to assess the quality of the rendering as sometimes the HTO signal associated with each sample approaches the low signal area and thus makes it more difficult to determine the original sample. When your analysis allows you to consider the result to be of good quality, the Filter Out Multiplied and Negative button allows you to eliminate the cells considered as duplicates or negatives, in this case 852 plus 225 cells. ShIVA then proposes to create a new project which will be in fact a subproject of the first project created. This will allow you to easily go back to the initial project to perhaps apply a manual demultiplexing in order to do new analyses. As you can see after applying the elimination, the double and negative do not appear in the table or in the UMAP. And the current project is indeed the After Demux project, while the old project can be accessed through the Project Select menu. Once the HTOs have been demultiplexed, you can move on to the quality control stage, which is the subject of another video.